Okay, so welcome to the last of your online labs. Since it's the last one, I will try and keep it brief and straight to the point. You are already sort of pros in this area because today we're going to be talking about ELISA, which is enzyme-linked immunosorbent assay, a very valuable diagnostic tool, which is similar to what you've been hearing on the news recently. So now, as I mentioned, some of this will be a review for you since we covered it in lecture, but it's especially important during the current state of the world. Now, what we're focusing on today is simulating diagnosis of infectious disease. And just to review, whenever you hear me ask, well, what are some diagnostic methods? There are three main categories that should come to your mind. There are the growth-based ones that we talked about, so things like the enteropleury test, or selective and differential media. There's serology, which is what we'll be focusing on today. And there are, of course, my favorites, the nucleic acid methods, things like PCR and microarrays. So today, focusing in on serology, that's something that you've been hearing all over the news lately. Serology is very important with diagnostics and studying infectious diseases such as viruses or bacteria, because serology focuses on antibodies and antigens. Okay, so we use the idea that antigens of, let's say, viral proteins or bacterial proteins get bound by very specific antibodies targeting them. We can use that concept to diagnose people, and you're going to see that as we talk about ELISA today. And what you'll be looking at is a simulation of using ELISA to detect an HIV antigen. Now keep in mind, even though we're going to focus on HIV today, ELISA can be used for detecting a lot of different antigens, okay? Any viral or bacterial protein, things like STDs, and as we mentioned in lecture, ELISA is even the concept of pregnancy tests. Okay, so it's a very valuable, powerful tool, but it's based on the concept of specific antibodies will bind specific antigens, and that idea that this is part of your natural adaptive immunity, which we discussed in lecture as well. So keep in mind the concept of adaptive immunity, the helper T cells activating the B cells, making those antibodies, and that binding is going to be the basis of the experiment today. So when we talk about ELISA, there are 4 general steps that you have to know, not only what the step is, but the function of it. Why do we do it? So the very first thing you do when you're performing an ELISA is you take your set of wells, and you'll see later that they also have paper strip versions, but we'll keep referring to wells. So you take your set of wells and you add a known antigen to every single one of those wells that you're going to work with that day. Now, when we say known antigen, if, for instance, today you are trying to diagnose HIV in your patients, well, the known antigen that you would put in each of those wells is an HIV protein, so the protein tag of HIV. So all of the wells get coated with that. Then normally what you would do is you would wash away any excess with detergent, and the detergent would also serve as what we call a blocking agent that you use in serology techniques such as Western blot and ELISA. And what the blocking agent does is, if you picture a whole bunch of wells, when you're putting your antigen in them, not every speck of that bottom of the well is going to be coated with antigen. So the blocking agent will fill in any of those extra gaps so that later on you don't get any non-specific binding and false results. Okay, but the key here is, first step, you are putting your pathogen antigen into each of the wells that you're going to be using. The second step is what we technically call the primary antibody. But what it will actually be in the actual experiment is your patient's serum. So you would take your patient sample now and put it into those wells where you had just added antigen. So now here is the important concept, the real core concept that you have to understand for ELISA. When you do this step, when you add your patient sample into those wells, there are two possibilities. If your patient is currently infected with, let's use today's example, HIV. If your patient is currently affected with HIV, then what has their body started to do? Well, their body is fighting that infection, and so it has produced specific antibodies to target HIV. Okay, because remember at this point, once your immune system is activated, those helper T cells activated the B cells, you have antibodies specific to HIV that have been produced. So if your patient is infected, then their patient sample has HIV antibodies, or antibodies against the HIV, I should say. So now when you put that patient sample into the well, where you have already put HIV antigen in, what will happen with your patient sample if they're infected? Well, the primary antibodies specific to HIV will bind to the antigen in the well. If they are not infected, what's going to happen? If they're not infected, there's no immune response. Their body has not produced any antibodies. So when you put their sample in that well, there's nothing to bind to the antigen. There's no reaction that's going to occur. You then add the third step. The third step now is what we call the secondary antibody. Always keep in mind, primary antibodies are specific. Primary antibodies will only bind to their specific matching antigen. Secondary antibodies, you have to remember, are general antibodies. Secondary antibodies will bind to any primary antibodies. They don't have specificity to one particular protein. Instead, they're built to bind to any other antibodies. So when you add this secondary antibody, if there is currently any well that has a primary antibody complex in it that's stuck because the patient was infected, the secondary antibody will bind to those primary antibodies. Now, the other aspect of the secondary antibody that's important to remember, not only will it bind to primary antibodies, but it's also specially built to have a form of detection attached to it. So a lot of secondary antibodies, what they do is they put what's called enzyme labeling on them. Specifically, they like to label them with HRP enzyme, like your kit today. HRP enzyme is horseradish peroxidase. That will react with our final step in a minute, causing a color-changing detection for us to see. So the secondary antibody in an ELISA is very important because this will bind to any of the primary complexes of the infected patient's samples, and it will have a form of detection on it. Okay, so remember those two important aspects of it. It binds primary antibody, or the patient sample antibody, and it has a detection on it. That brings us to the final step, which is when you add what we call a chromogenic enzyme substrate. And when you hear chromo, that usually means color. So chromogenic means color-producing substrate. This final step, which is adding the substrate, what happens is it reacts with that secondary antibody's HRP that we just mentioned a second ago. And what happens is that HRP from the secondary antibody will catalyze what's called the oxidation of the final substrate. That's a fancy way of saying that the secondary antibody's little HRP that's hanging on to it will break down the chromogenic substrate, the final reagent that you're adding in an ELISA, and it will cause a product to be produced that has color. Okay, so kind of like you've seen in some of the other experiments that we've performed this semester, like in transformation, when XGAL was broken down so that one of the products was blue. In today's kit, the final substrate will be broken down by HRP and produce a purple color. But when you look at pictures, let's say online or in various other labs, they use all different color detection. So some ELISAs will turn purple, some will turn pink, blue, green, yellow, whatever. All that matters is the fact that you will see a color change in an ELISA, and that tells you that that sample is positive, okay? It has the matching antigen or the specific pathogen that you were targeting. So keep in mind, if you see a colorless well, it will be negative. If you see a purple well in today's results, it will be positive. And the reason that it is positive is because you had put an HIV antigen in those wells. The patient's sample then had the matching antibody because they were infected. You then had the secondary antibody bind to that patient's primary antibodies. And that secondary antibody had HRP that oxidized the final substrate. And that caused the color change to be produced. Now you will notice that some of the wells might look lighter than others. And I'm going to want you in the post lab to analyze why you think that is. And what I want you to think about is, well, what's causing the purple color in the first place? And once you realize what would cause the purple color, the exact scientific, immunology reason for that purple color, well, then maybe you can start to think about why some will be lighter or some will be darker reactions than others. Okay, I'm not going to give you the answer because I want you to do some critical thinking in today's lab. Okay? So this is just a recap picture so that you can see each step. Like I said, the first step is where you put the antigen. So you can think of this as the suspected pathogen. So whichever thing you're testing for, if the ELISA is for HIV, this will be a piece of protein from HIV. If you're testing for COVID, this will be a piece of that virus. If you're testing for the flu, same thing. It'll be a piece of pathogen protein. Then the second step is you add the patient's serum. If the patient is infected, then their body has produced antibodies that match the pathogen you're testing for. So the patient's primary antibodies will bind to the antigen. You then wash out any excess and you apply the secondary antibody, which you note has detection on top, in this case HRP. So the secondary antibodies will bind to the patient's serum antibodies, and that HRP will then in the final step change that substrate, the final substrate that you add to an ELISA. So the HRP will break that down into colorful product. Now keep in mind, if your patient is not infected, then they don't have any antibodies in their serum. So you never get this complex able to be formed. Instead, you end up washing away those the sample. You wouldn't have any binding, you wouldn't see any color change. So the final product would be colorless. This is just to show you what we mean by the wells. So all of these little circles are individual wells that you would be pipetting into. I also want to note that nowadays they actually make what are called rapid strips. So basically they look like pieces of filter paper or even just regular old paper. You pipette into the circles and you will still see a color change for anything that is positive. And notice you always want to have your controls first. So even today's protocol, you'll have controls set up first, always a positive and a negative, to compare your samples to, and most importantly, to make sure that all of the reagents and the test is working accurately. Because when it comes to diagnostics, you never want to be the person who gave a patient the wrong information, either telling them that they were sick when they weren't, or telling them that they were not sick when they actually did have the particular pathogen. Okay, so you always need controls in an ELISA. Now, what I love about this experiment and this kit is that it gives beautiful real-life examples. So in today's ELISA, when you're analyzing your results, these are your actual patients that you will be testing to see which ones have HIV and which ones don't. And all of these are real-life scenarios that can lead to an HIV scare and the need to be tested for HIV. So patient A has just learned that her ex-boyfriend was diagnosed with HIV. Notice it says, although they often use protection when sexually active, not always, she is concerned that she could have contracted the virus from him. Now, patient B is current boyfriend of patient A. So even though he was unaware that, she has these exes, you never know who someone's ex could have been, he's now concerned because even though they're not sexually active, they have engaged in deep kissing, and he's concerned that there might have been some kind of exposure to HIV. Plus, he also wants to show support, so he's getting tested along with her. Patient C is a sad scenario, and it's one that I want you to all pay attention to because a lot of you are going into the medical field, either as nurses, PAs, or even working as EMTs right now, and this is a real possibility. Patient C was treating a bleeding car accident victim, but at the time didn't know that the person was HIV positive. And so after attending to that person's injuries, he then realized his glove was torn and his hand was cut. Okay, so you always have to be extra careful because even though you think you're protected with your PPE, things happen. So you always have to be very alert and cautious. So he has to be tested now. Patient D is a scenario that a lot of people are familiar with. Broke up with a boyfriend after learning about several lies he told her. She worries that he has not been monogamous and decides to be tested for HIV. Now, this is considered one of the scientific terms of fuckboy transmission of infectious diseases. Very, very common nowadays. So please be very careful. You never know what your partner is doing. You can think they're the most perfect person in the world, but sometimes they are lying. And unfortunately, she now has to be tested because even though she did everything right, unfortunately, she was with someone who was not doing everything right. You then have patient E, which is another scary scenario that many of you may not think about. Patient E is now happily married, has young children, has his whole life together, everything going great, and then boom, he gets a call from an ex-college girlfriend. Turns out she is HIV positive. She's not sure, though, when exactly she contracted the virus, and so now she has to contact all of her old partners just so that they can get tested as well. Because as we've mentioned in lecture, you never know. With viruses or infections like this, they go latent. And so he could have caught the infection from her if she had it back then, and he could simply not be showing symptoms yet. Or maybe she got it after she had been with him, so he doesn't have to worry in that scenario because she was not HIV positive at that time. You never know when it comes to these kinds of infections. The last one is a patient experimenting with drugs during college, and this one shows you, kind of like the EMT showed you, that viruses that are, normally seen as STDs are not only transmitted through sexual activities. So in the case of patient F, he may possibly be HIV positive due to drug use, sharing intravenous needles, and now he's kind of got to worry about the past coming back to get him. So these are all of the scenarios I want you to keep in mind. Look back at these to remind yourself later on in the experiment, once you figure out which patients are positive and negative, really look at the activities and what happened. But in reality, technically, any of these people could have gotten infected. And I know you may be thinking, well, patient B, all he did was some deep kiss, and I don't think he has it. but you actually don't know, because technically even things like deep kissing or oral sex, if by chance you just happen to have the worst luck in the world, like Bad Luck Brian memes, and well, you happen to have a cut at the right location during the right activities, and the other person, you never know what's going on with their body as well, things can happen. So it's not impossible. The statistics are lower, but it is not impossible. So any of these scenarios can transmit the infection. So in each of these next slides, I've given the protocol for you to review as you are analyzing your results, which will be on the final slide. Basically, what you would be doing if you were doing this in lab is you would have that plate with all of those wells that I showed you before, and you would be pipetting in the different reagents that we mentioned earlier. So that would include putting all of the antigens. So in this case, it would be antigen against HIV into each of the wells, then putting in each of the patient samples, but first putting in a positive and a negative control. And remember that those are very critical. When you go to analyze your results today in the post-lab, please be very careful. You may want to draw on your printed out slides to remind yourself where A1, A2, A3, each of these wells, what you're putting in them, okay? This continues the protocol, and it also maps out for you. So these are your 3 positive controls, your 3 negative controls, patient A, patient B, and so forth. And this will be the wells that you see in your results. So what I usually like to do is I draw a line around the 3 repeats, which I will also be asking you about in your results, why it's so important that you'll notice for each of the samples there are 3 wells. So every sample is repeated 3 times. And think about the purpose of this test and why it's so important to have repeats, especially since you are then going to have to tell someone whether or not they have HIV. And here's the final steps in the protocol for you to review. Again, when you look through the protocol, remind yourself of the 4 main steps of any ELISA. The antigens, the patient sample, which should either have primary antibodies or no antibodies, the secondary antibody, which is enzyme linked for detection, and the chromogenic substrate. So keep reminding yourself of what these things are, why you're doing them. And here you have your results. So notice that we only use the top of the wells. In this case, we did not use the full plates. So when you're analyzing, this top area matches the protocol's guidelines for what samples are placed where. And like I said, you'll notice that some of the results end up looking a little lighter than others. And you may want to ask yourself, well, what does that tell you about that patient sample? Okay, what could have happened to cause lighter versus darker reactions? And again, asking yourself, well, what exactly is causing these reactions? Okay, so I really love this experiment because it is real life examples and it's very applicable to, things that you will be encountering. So please kind of use this as one of the additional kind of lessons for yourself in terms of being very careful about not only things involving your patients, but about things involving yourself. And the decisions you make, no matter how old you are, can affect the rest of your life. So I know you may think like, oh, young, fun, what I do now doesn't matter because I'm young and my body can handle anything. Well, you're not going to feel that way down the line if, let's say, you want to have a family. And, well, what happens when these decisions that you make now catch up to you? Okay? So on that horribly depressing note, and I apologize for ruining all of your good times, let me know if you have any questions, if anything's unclear or you want me to review it, anything in Remind, just let me know. Good luck, and keep in mind that this is the last of the experiments or online labs. So next week is the final. And for that, my recommendation in terms of reviewing for it is, first of all, go to the announcement that has been posted and remind yourself the exact experiments that have been done this semester. So there are about 12 topics that have been listed out for you in terms of what this final covers. It's all of the semester-long experiments, so that includes the weeks that we were in person and the weeks that we were online. And like we've said so far, it will be all multiple choice. And so make sure that when you're kind of studying these things, you have the key points of the background information, but also analysis information as well. So for instance, for an ELISA, if you see purple well versus colorless well, well, what would that tell you? If you see a blackened agar, well, what does that tell you in an agar slant, a bioescalin test? What does that tell you about the result? Novobiasin disc without a zone of inhibition, what does that tell you? So don't just focus on background information, but be able to understand some results as well. Even things like the temperature experiment, the salt experiment. We had specific bacteria we worked with that should kind of stick in your mind. Staph versus E. coli in the salt experiment. Bacillus serratia E. coli in the temperature one. Okay, so review your handouts, your notes. Also review the quizzes that you've taken. Okay, you get to see your feedback on the online ones. We gave back the written ones. So review your quizzes, because that also helps remind you of some of the key concepts. And review your post-lab assignments as well, because that's a quick way to kind of review some of the key points. Notice that the online labs are very similar to how we have in lecture. There are things in bold, there are things in color, and there are actual questions written on the slides as well. Okay, so keep all of that in mind as your final review. Okay, so kind of threw that in as a review for the final. We're not going to have any other separate posts regarding a review. You have everything you need kind of mapped out for you already. And as always, don't hesitate to ask me any questions. Okay, send me remind messages as you are studying. Okay, thank you and have a great day.